My topic is viral diagnostics. Chili is one of the important crop where we can see biotic and abiotic stress. Of the biotic stress, see diseases are play important role. Disease caused by viruses are very destructive in nature. Of these 68 viruses species are reported. Of it, 45 species are from 8 genera, indicating these genera play important role for causing the disease in Chile. See, in the worldwide, you can see many number of viruses reported in the world. Among the India play important role for viral diversity. In South Asian country, you can observe the diversified viruses associated with Chile, indicating this there is a huge problem from the viruses associated with Chile. They can see different symptoms associated with Chili, either chlorotic or necrotic or mosaic or leaf curl associated with begum viruses, and also the browning, it is due to the brown fruit rugose virus. Indicate this important virus disease in Chile, the Chilli leaf curl disease, in which is transmitted by white plague, where this Chilli virus, yesterday there was huge discussion regarding Chilli, as Ashpetapan from worked under guidance of Dr. Krishna Reddy sir, indicated the occurrence of more than 14 species of begum viruses associated with Chilli in India. This is a destructive disease, and this both biopartite and monopartite viruses associated with this disease. Next is important, next to tomato leaf curl virus is Chilli vine mottle virus. Is extensively work done by Lakshmana Reddy under guidance of Dr. Krishna Reddy for his PhD thesis. It is the first report from the India indicating that it is a destructive nature. For his PhD, he got Governor Award under guidance of Krishna Reddy sir. Another important is you can see pepper mottle virus is infected Chili associated in India, and Jacqueline yellow vine mosaic also associated in the capsicum in India. And cucumber mosaic virus also third important viral disease associated with Chili and capsicum reported by Krishna Reddy's team. Then you can see another orthotospovirus, other different species, capsiclorosis virus, and also groundnut bed necrosis associated with Chili from India. Like this, other minor viruses also associated with cucumber mosaic along with your chlorosis virus. This is a complex virus associated with Chili from India. These tobacco mosaic virus or the tobacco virus also is destructive in nature. These are extensively transmitted through seed-borne and mechanical. The pepper mild mottle virus also is an emerging problem and also seed-transmitted virus. There is some virus, minor virus like pepper cryptic virus and tobacco stick virus also reported on Chili from India. This why, after knowing all these, why diagnosis play important role. Accurate diagnosis combined with the early detection of plant virus is critical for management. Especially for plant quarantine, diagnostics play important role for detection for the restriction of the entry of the pathogen. These diagnosis or detection tools play important role for sampling and testing the sample for epidemiological, and they play important role surveillance and monitoring of the viral disease. See, to detect diseases, there is a different tools are used. You can see usual symptoms, and also through transmission, indicator host, electron microscope, immunosorbent, and some nucleic acid-based techniques also are exploited. And nowadays, people are going for nanosensor artificial intelligence also. See, these are based on symptoms. We can detect few virus, but it's very difficult. And we can use transmission to detect the presence of virus, either grafting or through seed. This transmission will play important role for characterizing the virus. Indicator host, where you can a biological host for detection of the pathogen. Another electron microscope, where it has ruled the physical characterization of the viruses initially. Then based on serology, we can detect the virus. There is the recruitment of antibody for detection of the virus. There are the different ways taken antigen preparation, either the conventional purification through the propagation method, advanced using a recombinant DNA technology. We have used the recombinant DNA technology to develop the polyclonal antibody against tomato leaf New Delhi virus is a destructive pathogen associated with different crops. And once if you construct and purify the protein through recombinant DNA technology, inject into the rabbit and develop antibody. Through this, you can establish different serological tools. Once you have antibody, you can, there are several workers have worked to characterize through a serological, and also antibodies are produced in the virus-associated chili. Then another very important quick method where you can exploit the field level, that is the strips, how pregnancy test 10 and the COVID time, it has ruled the world for diagnosis. This is the quick, fast, and also field deployable. Then disadvantage, there is requirement of specific antibody, there is a cross contamination, then it cannot be used for large scale indexing of a virus. Then the Ashwatthapa used for PhD for detection of 12CV, NDV, immunostrips in chili, and there is a development of immunoglow strip for the detection of cucumber mosaic virus. Another, these molecular tools, because serological tools have their own disadvantage, molecular tools have advanced in nature, as PCR tools discovered by Carey Mullis revolution the diagnostics also. This where Ashwatthapa used the diagnostics for detection of the chili leaf curl virus in different parts of the chili, including seed. That means it is seed born in nature also. And also he has used the detection of chili leaf curl virus in asymptomatic samples also. RT-PCR also revolutionized the detection, where the RT-PCR are exploited for the RNA virus and the viroid, because their genome is RNA in nature. This RT-PCR was used to detect the seed-borne nature of the pepper mild bottle virus, and several workers have used this RT-PCR for detection of seed-borne nature of the virus associated with the chili and pepper. Another important is immunocapture. In case of some interfering plant substances interfere in the amplification, to avoid this DNA extraction or extraction of the nucleic acid, we can capture the antibody through, and then we can use that as a template and use for the immunocapture can be used for detection of a virus. Then multiplex also can use for indexing the virus associated different crops. Then morely accepted for the quarantine is real-time PCR, when in the more advanced of a PCR, you can low quantity of the concentration of a virus you can detect, is more accepted with respect to the detection and also quarantine importance. You can see here, Ridge has used the seed-borne nature of the brown rogus virus associated pepper and indicated this more sensitive. Ashwatthapa used the detection of chili leaf curl virus in the different parts of the plant. Then the concentration is more in case of leaf as compared to seed. He has also indicated the presence of virus in the seed also through real-time PCR. Another advanced tool is the loop-mediated, the field-based technique. In order to avoid this, each of the equipment like a PCR, it is a field-based and also rapid technique. This is what we have used the tomato leaf curl noodle virus. We have standardized the LAMP techniques for detection of. Advantage of this technique is to avoid the running of the gel. You can using a intercalating dyes. Like that, Ashwatthapa used the detection of the chili leaf curl virus using this intercalating techniques through LAMP. Another advanced tool is RPA. where you can avoid within the 10 minutes, 10 to 50, 20 minutes, you can detect the pathogen. It is isothermal amplification. The Devi and core course use this RPA for detection of the Chilli vinyl mottle virus and standards, indicating these techniques can be used for the extraction of the detection of the virus in the sap also. Recently, Anusha has established system RTA for detection of the cucumber mosaic virus also. Another now science advance, we have to advance. There is a detection tool coupled with the CRISPR, where isothermal application methods are coupled with the place. This is a field-based techniques. There is recently there are two reports indicating the CRISPR coupled amplification tools for detection of the viruses. Next generation has revolutionized the discovery of the novel virus without the information, the antibody and prior knowledge of the virus. This is more useful for the discovery of novel virus and also quarantine important pathogens also. This is the very important here. The analysis part is very important. Knowledge is required to analyze the sequence. Recently, there is the Reddy and co-workers from US, Bangalore, has established the NGS for the detection of, they use the, I'm also part of this work, 7 virus associated Chilli with a diverse, indicating these NGS can be exploited for discovery of novel viruses also. They also developed the, sorry. Jump again. Back. 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 Yeah, they established a RT-PCR and real-time RT-LAMP for the detection of these identified viruses. The next. See, in international trade, the quarantine play important role. Viral disease are among the most serious constraints where you can chili production causing yield loss, food deformation, quality, and a very important trade restriction. Many of these are seed transmitted, quarantine regulated. The accurate detection and diagnosis play important role. Reliable and early detection and diagnosis of pathogens are critical for plant quarantine, seed health testing, and certification programs. In this regard, as a part of our university, we have received many samples from seed companies, the regional plant quarantine station, Bengaluru and New Delhi, Mumbai, Chennai, and Department of Agriculture, and many seed companies through themselves or through some agencies. About 237 samples during 2023 and 535 samples during 24-45. We have received for detection, it has the quarantine importance, indicating the chili and tomato play important role for diagnosis for quarantine importance. Look at this 76% sample among the states, the tomato and chili during 2023 and 60% samples among the tested because the number of samples, number of crops increase because of surveys. Indicating this, the virus during 24-25 for the detection is virus is of 34. You can see we have given a service during 23-24 through our diagnostic lab. 43 companies are benefited for detection of the pathogens. And during 23-24, 70 companies we have given service for the detection diagnosis of the quarantine important pathogens. Then what were the, after sampling and testing, more than 34 pathogens are associated with viral in nature, 9 are viral and bacteria 10. These all are sample received for quarantine importance. In this regard, see, accept a protocol for detection of quarantine plant pathogens, either virus or viral, PCR for DNA virus, RT-PCR for RT-PS or real-time PCR for RNA viruses and viroids. Then bacterial fungal pathogens, selective media isolation, PCR amplification and sequencing. We are extensively using a PCR for DNA virus and RT-PCR for real-time PCR also is required. In the record, we have established a system where we can see extraction in the nucleic acid, especially viral and standardized for the protocols of RNA, and there is a tool for the real-time PCR and also PCR for DNA and RT-PCR for RNA. Once if you get the results, then you can issue the report. In this records, we have standardized the protocols for some positive controls. Positive controls play important role for detection of the virus. We can see a cucumber mosaic virus, degenerative promise where it can generative in nature, and tomato brown rugose virus is reported in tomato, and also some samples in capsicum also we have so found positive. Like that, we have many positive controls we have established, and Zikini mosaic virus, and also brown rugose, cucumber green mottle virus. During detection of any sample, positive control play important role in order to see your virus is amplifying or not. Like the XCV party virus and also watermelon bednacrasis, we establish positive control for detection of virus. And also we can see the many number of viruses, we have established a positive control for detection of a pathogen associated quarantine importance. And if we don't know any positive controls, what you have to do? We have to do positive, what is positive close. In this regard, we have synthesized the gene of the virus or virus. Next, we have to amplify. Then we have to ligate the vector. Then once if you ligate, you have to confirm the clone. Then we can use. We have used some positive color. This is supported by Prime Molecular Science Private Limited, where you can see the plasmid isolation. Then colony PCR. Once if colony PCR done, we have to extract the plasmid and check the presence of our insert. That plasmid can be used as a positive control for detection of any virus or viral, any pathogen, because sometimes we don't have any pathogen in country. But that time we have to use some positive control. In this regard, we have started working on positive control, developing a positive control for detection purpose. Finally, I should conclude, chili is one of the important decidual crop. Viral disease play important role for reducing the yield and quality. But the wise of disease planting material is a crucial role for the effective management of sustainable cultivation. Several diagnostics are available for detection of the plant pathogen. Among these, PCR, RT-PCR and real-time PCR, that is qPCR, are most reliable, sensitive, widely accepted method for detection of viruses, viral, in which quarantine important. Finally, I should acknowledge and thank all my project staff who are associated testing the sample. I should grateful to my mentors who taught me basics of virology. Professor Mundiappa is associate is a lifetime, more than 45 years in virus, and Ragaswamy is my mentor, and my postdoc mentor is Professor Durga, where I learned molecular biology. I should thankful to Dr. Krishna Reddy sir, where I learned basics of virus during master. Still he is my mentor. Thank you one and all.
Thank you very much. That was a very nice presentation. I have received one comment in my WhatsApp. Diagnostics is good, but what about management? This is nobody is talking about, I think, I don't want to name the person, but he said, what about management? This is this thing that has come. I know there are segments. Your focus was on diagnostics. Management is altogether a diversified subject, but I would like you to add a small comment with as a reply, how diagnostics contributes to management.
Sir, prevention is better than a cure, sir. If you use prevention is better than a cure. Once if you use the disease material free from disease, we can 50% can avoid. For example, now there is a system where DBT accredited lab, they have made a rules. You have who are purchasing a banana, they have to test in the DBT accredited lab that mother culture should be free from diseases, or especially virus or virus. Once if use your prevention is better than use, at least we can reduce some. Now, for example, you don't know diagnostic tools. If you are exporting, importing, then how we can diagnose? Most of the viruses like that only spread all over India, all over country, all over the world. Prevention is better than cure. Thank you, sir.
Any other questions from the audience?
Check. A farmer brings seedlings from the nursery. Do you have a strip or some tool? You should just place it on the leaf. It should tell virus is there or not. And how cost effective it is. If we can answer these two, we can avoid so many problems. Thank you.
With respect to strips now, sir, what Harish sir has said, that type of field-based strips can be utilized, sir. But that there is a requirement of antibody and establishments.
There are two things, Dr. Harish, I'll answer in his defense. There are a number of diseases where with a normal experience, a good virologist will be able to tell. There are certain cryptic viruses which only need a diagnosis, without which it will be, sometimes there will be mixed infections. Supposing I am exporting seed material, okay, and I give you the seed to say whether it has got a virus or not with no symptoms, you have to go through a molecular diagnosis. Supposing I am importing a material from abroad, I cannot blindly import a material without a molecular diagnosis. So there are dimensions what we are talking, which you are talking, but from a farmer perspective, I also support what Dr. Harish is telling is, for most of the common viruses, at least about 5 or 6 which you consider are most prevalent, if you can come out with a cost-effective strip, thereby what happens is extension will be. That is what he is probably.
Yes, sir. Definitely will come out, sir. Definitely we are planning, sir. Planning any industry who are working. Yes, yesterday sir was telling, we'll come out with some outcomes, sir.
Heli Harish.
Sir.
Nursery is not certified, and we do not have laboratories to certify it is disease free. At least a block level we do not have, sir.
Sir.
Farmer is not knowing whether I am using disease free seedling or disease seedlings. All virologists, it is my sincere submission to the house, we should develop low cost field based diagnostics which farmer can afford and use it. He should be able to himself tell whether it is disease free or not. This is the expectation.
Before I also tell about virologists, I also tell about all agronomists, horticulturists. Let all your polyhouse doors not be opening to the road.
Sir,
you are most of the material that you are generating, most unscientific polyhouses, most open to the door. Anybody comes, the door is open, and then you expect the virus-free seedling. You know it. I'll not go into discuss. I'll give it to Krishna Reddy for.
95% it is open field cultivation, not polyhouse.
No, seedlings. I'm talking seedlings. Dr. Krishna Reddy, please.
I would like to answer all the questions which are regarding to diagnostics. The diagnostics always not help for the farmer. Please remind, this diagnostics for viruses, not for the farmer. The diagnostics which are to be used for farmers, even if the disease is known to the farmer, what he will do?
you cannot use.
You cannot use. Wait. The annual crops, seedlings raised, virus is not there. The moment it is kept in the field, all viruses will come. So diagnosis has no meaning in that. The seedling, disease free, has no meaning in nursery crops. But propagating material importance is there in perennial and ornamental crops. Now in India, we are establishing clean plant program where all the perennial crop certification is done through this, and the system is establishing now. Already we have in banana, now it is extended to 10 fruit crops, where the farmer, the planting material takes years of establishment and he cannot destroy the crop. So there plays important role in diagnosis. For annual crops, seedling raising is important. Nursery production is important. He has to manage disease-free, scientific, is the production manager, nursery people, not the farmer. But we can, I gave this for at least 5-10 farmers meetings in Karnataka and in 10 farmers meeting in Maharashtra. Government should take act now on seedling production of the nurseries, should categorize A, B, C groups. A means excellent, where scientifically produced, disease-free they are producing, that has to be accredited. B is moderate, C is not eligible. If that category is given, farmer is benefited, but all your diagnostics is not going to help. Yes. It is the service we have to provide, establish a system that helps the farmer. That is my...
Thank you, Doctor. I want one minute. I wanted Dr. G.S. Prakash. He was very seriously discussing, and I know that he comes out with serious comments.
No, I'm familiar with what Dr. Krishna Reddy was telling. My only one question was, your first statement was little confusing, because see, in case there is a infection after going into the field, what is the necessity to check in when it comes out of the nursery? Dr. Krishna Kumar insisted that we should see that we produce disease-free plant material as a starting healthy plant material, which is important now. So if you say there is a lateral entry as soon as you transplant, we need not to bother. No. Entire tissue culture, as Dr. Prabhakar is telling, stands on this premise that we start our program with a disease-free, healthy plant material. That let us accept. And lateral entry, yes, there are many other issues.
So I am clarify that again, clarify. That is for perennial crops. For annual crops, let me tell, annual crops, seedling production, especially solanaceous crops, nursery persons, that is what I told. We have to certify the nursery, not the seedlings. Sir. Nursery person who is producing production system to be certified. He has to be A, B, C, D groups.
You are again justifying what you say.
We'll come to this. My only this thing at the last statement, I would like to, Dr. Krishna Reddy will continue the discussion.
Yes.
Okay.
Sir, one submission is a policy decision should be taken. All nurseries which are supplying the seedlings to the growers, it should go as certified. There is no regulation on nurseries right now in Karnataka or elsewhere, I don't know.
That is what the requirement is, that one only.
1 minute. We also want those backbenchers who are there. They may have serious issues, but we should give them opportunity also. They come forward. Anybody? Sir. Very pointed question. Heli, please.
Sir, again, my question is related to Dr. Sir. So regarding
that you discussed during tea hour.
Yes, sir. That is
that day you discussed during tea hour. Any new questions?
Sir, there are some nurseries in Maharashtra regarding pomegranate. They are having some rules and regulation from the government of Maharashtra. Likewise, are we having any rules and regulation for the establishment of nursery?
No.
If you are talking, 99% of the nursery structure itself is unscientific.
Right.
And I, for one, slightly differ with Krishna Reddy that the day you involve the government, you can be certified that inefficiency, corruption, all these things are going to creep into this. Karnataka, Kerala may be better. What about Bihar? What about UP? What about many other states? I feel that a time has come when we have to follow the Maharashtra model. Take the leadership to the farming community. They will do a very scientific job. They know what exactly to do. Other states should gear up to that kind of a model. Otherwise, in states like, I don't want to call those states, expecting the government to act on these things.
Sir, my request on behalf of the House is, we will push to the government that
we'll come out with a policy.
We have to raise the rules and regulations to avoid the disease spread from the nursery itself. Because the source is nursery only, from there only spreading viruses and disease,
with both of you, one small clarification.
Yes.
What happens when it comes to the field is not the question, because he is also right.
Yes.
The removal of the primary source inoculum material itself is a great service in the secondary spread of the virus. Yes. So what happens is, even though you diagnose it in the field, The first plant, if you diagnose it as infected and remove it, a lot of lateral spread can be prevented.
Definitely it will
So
be.
it's a crop by crop, issue by issue, place by place, virus by virus.
Yes.
So we'll discuss it more at detail when
Sir,
we
in-
go through the chili leaf curl virus management.
Thank you, sir.
Sir,
I
in
think
so-
we are running out of time.
One submission, sir.
Yes.
Sir, here, sir.
Where are you?
Here only, sir.
Okay.
From recommendation from this conference, because when the 2023 outbreak of a tomato leaf curl in tomato again in Kolar, we are given directions to the government of Karnataka to introduce the Nursery Act. We can also give a recommendation from this conference also, sir.
We can give the National Biosecurity Act recommended to the Parliament in 2012 is still hanging in. Until today, we don't have National Biosecurity Act. So I was just drawing parallels to that as to how far this country acts on recommendations. Leave apart that. What I would only request is, as Dr. Tikku said, please feel free to use the bowl, drop your suggestions. I am assure you that each and every suggestions of yours will be answered. Thank you very much.